Okay, guys, our today's topic is digestion of fats. We are discussing digestion of physiology. We discuss digestion of carbohydrates and digestion of proteins. Now, today's topic is digestion of fats. So, these fats they are also digested by the help of some enzymes. Those enzymes which are involved in digestion of fats they are known as esterases. Why we call them as esterases? Because these enzymes they break the ester bonds between glycerol and fatty acids. So that is why we call them as esterases. The most important esterase which is working in our body is the lipase. So lipase is an esterase which is involved in digestion of fats in our body. So this fat digestion, it actually starts from stomach. As I told you, in stomach there is a gastric lipase, but that gastric lipase is not able to digest a lot of fats because these fats they require emulsification, and after emulsification they are properly digested. So there is very little or no emulsification in the stomach, and this gastric lipase could not digest fats up to a greater extent in stomach. So actual site of fat digestion, where maximum fat digestion occurs, that is the intestine, small intestine. So the main site of fat digestion is small intestine. In small intestine, bile is released. Liver releases bile in small intestine. This bile, it contains bile salts in it. It has got bile salts in it. There are two main bile salts: sodium glycocholate and sodium taurocholate. And sodium taurocholate. So they are the most important bile salts. So these bile salts, they are involved in the emulsification of fats. They cause emulsification of fat. Emulsification of fats. So what is emulsification of fats? They break these bile salts. They break large fat globules. Large fat globules into small fat globules. Into small fat globules. So first of all, how they break these large fat globules into small fat globules? These bile salts they reduce surface tension surface tension of fat globules. So by reducing surface tension, they break large fat globules into many smaller fat globules. So this conversion of large fat globules into smaller fat globules by the action of bile salts, this is known as emulsification of fats. This is known as emulsification of fats. So what is the benefit of this emulsification of fats? So how does it benefit? So you know large fat globules they have got lesser surface area as compared to their volume. If this is a large fat globule, this large fat globule, it has got a greater volume and its surface area is lesser as compared to its volume. So I will give an example. You will calculate it yourself. These are two fat globules. This is large. It has got a radius of 2 centimeter. And this is a smaller fat globule. It has got a radius of 1 centimeter. So formula for area of a sphere. You can calculate the area of a sphere by the formula 4 pi r square. You can calculate volume of a sphere by the formula 4 by 3 pi r cube, right? So if you will calculate area and volume of this larger fat globule, you will definitely observe, and this smaller fat globule, you have to calculate area and volume of these two fat globules. This is larger fat globule, this is smaller fat globule. When you calculate area and volume of this larger fat globule and area and volume of smaller fat globule, you will come to see, you will notice that this larger fat globule, it has got surface area volume ratio. It is surface area as compared to volume will be less. In case of smaller fat globule, when you calculate surface area volume ratio, you will see with the decrease in volume, there is a proportionate increase in the surface area of the smaller fat globule. So emulsification of fats is necessary so that surface area ratio as compared to this volume increases. So that surface area increases and volume decreases. So by increasing surface area, lipases, fat digesting enzymes, lipases, they get more surface for action, and they digest the fats quickly. Without emulsification, lipases may digest the fats, but it will take a longer time. So when we emulsify the fats, when we break large globules into smaller globules, it increases surface area as compared to the volume. It increases surface area as compared to the volume. So lipases, they get more surface area for action, and this helps them to digest the fats quickly, right? So for digestion of fats, emulsification of fats is very essential. And this emulsification of fats, it is carried out in small intestine by the help of bile. In bile, there are bile salts, and those bile salts, they reduce the surface tension of this large fat globule. When the surface tension is reduced, this large fat globule automatically breaks into smaller fat globules. This increases surface area as compared to volume, and lipases get more surface for action, and they digest these fats very quickly. So fat digestion starts in the small intestine. In the small intestine, two juices are released. In addition to bile, two juices are released. In small intestine, bile is first released. You now know what is the function of bile. It is involved in emulsification. It emulsifies the fats. Then pancreatic juice is released. Pancreatic juice is released in small intestine. And intestinal juice is released in small intestine. Intestinal juice. Right? This pancreatic juice, it contains pancreatic lipase. It contains pancreatic lipase in it. And intestinal juice, it contains intestinal lipase. Intestinal lipase in it. So when fats reach into small intestine, first of all, they are emulsified. After emulsification, now these fats are acted upon by pancreatic lipase and intestinal lipase. They work together and they break. They start digestion of fats. How they digest the fats? So as you know, all the fats which we consume, they are triglycerides. So our dietary fats which we consume, they are triglyceride fats. Triglyceride fats, right? They are triglycerides. So these triglycerides, they are acted upon by pancreatic lipase and intestinal lipase in a stepwise manner. So pancreatic lipase and intestinal lipase, they change these triglycerides first into diglycerides plus a fatty acid. Plus a fatty acid. If you remember the composition of triglyceride fats, they are attached to a glycerol molecule. To this glycerol molecule is attached three fatty acids. One fatty acid, another fatty acid, another fatty acid. So three fatty acids are attached to a glycerol molecule. So when one fatty acid is removed, only two fatty acids remain attached to glycerol. So this glycerol molecule along with its two fatty acids. This is known as a diglyceride fat. So in the first step, pancreatic lipase and intestinal lipase, it removes one fatty acid from triglyceride fat, converting it into a diglyceride and releasing a fatty acid. Then further, these diglycerides by pancreatic lipase, and intestinal lipase, they may be changed into monoglycerides plus one more fatty acid. They may convert them into monoglyceride and one more fatty acid. So one more fatty acid will be removed. So when one more fatty acid is removed, glycerol and one fatty acid, it becomes monoglyceride. And this one more fatty acid is removed. Then monoglycerides may be converted into by pancreatic lipase and intestinal lipase. In next step, into glycerol plus one more fatty acid. So at the end, glycerol and three fatty acids may be released. But you should keep it in mind that fat digestion is never complete in our small intestine. Fat digestion is never completed in our small intestine. So what may be the end products of fat digestion? Because fats are not fully, they are not completely digested. Then what may be the end products of fat digestion? As we discussed about digestion of starches, I told you starches are completely digested and the end product of starch digestion is monosaccharides, right? Then I told you about protein digestion. So when proteins are fully digested, their end product is amino acids. Similarly, now fat digestion. The end product of fat digestion, they vary. The end product of fat digestion may be diglyceride plus one fatty acid. If fats are more digested, then the end product may be monoglyceride plus two fatty acids. Then the end product may be monoglyceride plus two fatty acids. If they are fully still more digested, then the end product may be glycerol plus three fatty acids, right? So the end products of fat digestion, they may be diglyceride plus fatty acid, or it can be monoglyceride plus two fatty acids, or it can be simply glycerol plus three fatty acids. So end product is not always glycerol and three fatty acids. This is not always. So we say end product of fat digestion may be diglyceride plus fatty acid, monoglyceride plus two fatty acids, or glycerol plus three fatty acids. Any one of the product may be formed. Or sometimes some portion of the fat is converted into diglyceride and fatty acid. Some portion of it may be converted into monoglyceride and two fatty acids. And sometimes glycerol and three fatty acids may be produced. So end product of fat digestion can be any one of these substances. Diglyceride plus one fatty acid, monoglyceride plus two fatty acids, or glycerol plus three fatty acids. Because fat digestion is not usually completed in our small intestine. So that is the digestion of fats, right? Now, after digestion, these fats are to be absorbed. All these digestive nutrients, they are to be absorbed. So for absorption of digestive nutrients, so we will discuss now absorption. Absorption of nutrients. So what is absorption? So we may define absorption as the process of transport. Absorption can be defined as the process of transport. It can be defined as the process of transport of nutrients from the lumen of small intestine into blood circulation. Since the site of digestion and absorption is small intestine, that is why I wrote small intestine. The process of transport of nutrients from the lumen of small intestine into blood circulation. This process is known as absorption. How this absorption occurs? This absorption may occur by two methods. One is known as passive absorption. Another is known as active absorption. This passive absorption, it is an energy independent process. It is a physical process. It is a physical process. It is energy independent. Energy independent. Dependent process. In passive absorption, no energy expenditure occurs. Our body does not make expenditure of energy for absorbing nutrients. So they are absorbed by physicochemical process, physicochemical method, right? So it is an energy independent process. It is an energy independent process. Whereas this active absorption, it is an energy dependent process. It is energy dependent process. For active absorption, our cells have to make expenditure of energy. Our cells spend ATP. And this hydrolysis of ATPs, it releases energy. By the help of that energy, our cells can carry out the absorption of molecules. So which nutrients are absorbed by passive transport? Nutrients like mineral salts, mineral salts, which are water soluble. They may be absorbed by passive transport. Water is absorbed by passive transport. Vitamins are absorbed by passive transport. Water soluble vitamins. They are absorbed by passive transport. So by passive transport, we are only able to absorb water soluble mineral salts, water, and water soluble vitamins. They are only absorbed by passive transport. So in passive transport, there is diffusion and osmosis. So in diffusion, there is movement of molecules or ions from the region of higher concentration to a region of lower concentration. What is osmosis? Osmosis is the movement of molecules or ions from the region of higher concentration to region of lower concentration through a semi-permeable membrane. By osmosis, only water is absorbed. And mineral salts and vitamins, they are absorbed by diffusion, right? So mineral salts, water, and vitamins, water-soluble vitamins, they are absorbed by passive transport or passive absorption, right? Whereas other nutrients, the main nutrients like amino acids, glucose, fats, they are absorbed by active transport or active absorption. So in active absorption, or we also call it as active transport. Nutrients are absorbed. By the help of some carrier proteins. Those carrier proteins, they are generally known as permeases or translocases. So in active absorption, nutrients are absorbed by the help of some carrier proteins which are present in the plasma membrane of the cell. Those proteins are generally known as permeases or translocases, right? They help in the absorption of nutrients, some important nutrients. So there are transporters. These proteins may also be known as transporters. So these permeases or translocases, they are actually transporters. which transport the nutrients across the cell membrane. So these transporters, they may be of different types. One is known as uniporter, a transporter molecule which allows movement of ions or molecules in one direction only. That is known as uniporter. Another is known as antiporter, and another is known as symporter. So there are transporters of different types. One is known as uniporter. It allows only the movement of ions or molecules in only one direction. For example, this is a transporter. It is present in the plasma membrane. It allows only movement of sodium or potassium in one direction. In the cell membranes, there are sodium gates, potassium gates. So these sodium gates and potassium gates, which allow the movement of sodium ions or potassium ions. Sodium gates, they only allow movement of sodium ions. Potassium gates, they only allow the movement of potassium ions, right? They are uniporters because they allow the movement of only a particular single ion across it, right? So they are known as uniporters. Uni means single, right? So these sodium gates or potassium gates, sodium gate, potassium gate, which are present in the plasma membrane of the cell. They are special proteins which allow the movement of sodium or potassium ion through them, right? They are known as uniporters. Then we have got antiporter. Then we have got an antiporter. Antiporter is a transporter protein. It allows the movement of one molecule in and simultaneously takes one ion or molecule out. Suppose it takes in calcium, and in turn of calcium, it takes out sodium. So this is sodium calcium antiporter. It is sodium calcium antiporter. It transports. It transports calcium ion into the cell, and in exchange of calcium ion, it takes out sodium. So this is known as sodium calcium antiporter. So in antiporters, two ions, they are allowed to move, but in opposite direction. Calcium enters into the cell cytoplasm, and from cell cytoplasm, sodium is taken out. So this movement is known as, this transport is known as antiporter, right? Similarly, there are symporters. Symporters are those transporters which take two ions or molecules simultaneously into the cytoplasm of the cell. This is the cell membrane. It is located in the cell membrane. This is lipid bilayer. In this lipid bilayer, we have got this protein. It takes sodium and along with it also takes glucose, right? So it makes the movement of sodium from high concentration to low concentration. So sodium is diffusing from high concentration to low concentration. But along with this sodium, glucose molecule may be taken from low concentration to high concentration. So this transporter protein, it is known as SGLT1. There are many different transporters. So we call this as SGLT1. Sodium glucose, sodium glucose transporter first. So it is taking sodium, this transporter molecule. It allows the movement of sodium from high concentration to low concentration. So sodium is diffusing from high concentration to low concentration. But along with this sodium, it also takes glucose from low concentration to high concentration. So these sodium glucose transporters, they ensure that whole dietary glucose is absorbed by the cells, intestinal epithelial cells, right? So we call them as Symporters. So these Symporters, they are essential in the absorption of very useful nutrients. There is another Symporter that is known as sodium amino acid Symporter. Sodium amino acid Symporter. Sodium amino acid Symporter. That means along with sodium it takes amino acids. There is another that is known as sodium iodide Symporter. Another is sodium phosphate Symporter. Similarly, there may be sodium potassium chloride Symporter, right? So these Symporters, they ensure that all the nutrients are absorbed by the intestinal epithelial cells, right? And in their transport, they may make expenditure of energy. Right? So that is why we call it as active transport, right? So active transport is brought about by the help of some protein molecules. Those protein molecules which are involved in active transport of these nutrients from lumen of small intestine into the intestinal epithelial cells, they are known as permeases or translocases. So we call simply them as transporters. Then we divide these transporters into uniporters, into antiporters, and into symporters. Uniporter allows the movement of a single ion through it. It takes either sodium ion through sodium gate or it takes potassium ion through potassium gate. Then we have got antiporters. Antiporter takes one ion in and one ion out. So in exchange of the ion which it takes in, it splits out another ion. For example, this sodium potassium antiporter, right? If sodium is taken in, potassium is secreted out. If potassium is taken in, sodium may be secreted out, right? Then we have got sodium glucose transporters. These sodium glucose transporters, they are transporting sodium. Along with sodium, they also transport glucose. They are carrying sodium from high concentration to low concentration. So sodium is moving down its concentration gradient, right? And glucose moves along with this sodium across against its concentration gradient. Glucose moves along with the sodium against its concentration gradient. That means glucose molecules, they may move from low concentration to high concentration, right? Sodium is moving from high concentration to low concentration, down the concentration gradient. And glucose moves against the concentration gradient from low concentration to high concentration. So this ensures the complete absorption of glucose from the lumen of small intestine, right? So that is about the absorption of glucose amino acids. Now, fatty acids, the end product of fat digestion, how that is absorbed, how fats are absorbed. For that purpose, there is another special mechanism. Fats are absorbed in a different way. So, as I told you, the end product of fat digestion, may be diglycerides, monoglycerides, and fatty acids. And also. So, end product of fat digestion may be diglyceride plus fatty acid, monoglyceride plus two fatty acids, glycerol and three fatty acids, right? So these end products of fat digestion, they are absorbed in a different way. So these digested fats, diglycerides with these two tails, this is a diglyceride, monoglycerides with single tail, only the tail. This is fatty acid, and this is the glycerol. So this represents a diglyceride. I'll show it here. This represents a diglyceride. This represents a monoglyceride, right? This represents only this line. It represents fatty acid, and this dot represents the glycerol. So these end products of fat digestion, diglycerides, monoglycerides, fatty acids, glycerol. Since glycerol can directly diffuse through the cell membrane, glycerol is an alcohol. It can directly diffuse through the cell membrane. So these fatty acids, monoglycerides and diglycerides, they cannot pass through the cell membrane directly. They get surrounded by some bile salts. They get surrounded by some bile salts and phospholipids to form a water-soluble colloidal aggregate. These diglycerides, monoglycerides, and fatty acids, they get surrounded by bile salts and they get surrounded by phospholipids. And they form a colloidal aggregate. They form colloidal aggregates known as micelles. They form micelles. Then these micelles, they come in contact with, they migrate into the villi of the intestinal epithelium. So they come in contact with the villi of intestinal epithelium. As they come in contact with the villi of intestinal epithelium, these villi of intestinal epithelium, they take them as small liquid droplets. So they take them in by the process of pinocytosis. These intestinal epithelial cells, they make invaginations. And these micelles, they fill in these invaginations. Then these invaginations, they get cut off from the surface of the intestinal villi, and they enter into the cytoplasm of the cell. This is an intestinal epithelial cell. This is intestinal epithelial cell. So when micelles come in contact with the epithelium of these intestinal cells, they make invaginations, and micelles aggregate in those invaginations, and these invaginations are released into the cytoplasm of the cell. So these small liquid-filled vesicles, they are known as pinosomes. So intestinal epithelial cells, they take these micelles in the form of pinosomes, right? And this process is known as pinocytosis. Then this pinosome, it joins with the endoplasmic reticulum of the intestinal epithelial cell. In the endoplasmic reticulum of intestinal epithelial cell, these fats are again combined. Diglyceride combines with a fatty acid to form a triglyceride. Monoglyceride combines with two fatty acids to form a triglyceride. Similarly, glycerol combines with three fatty acids to form a triglyceride. So the intestinal epithelial, in the cytoplasm of intestinal epithelial cells, these pinosomes, they join with the endoplasmic reticulum, and in the endoplasmic reticulum, there is a resynthesis of triglyceride fats. So in the intestinal epithelial cells, these diglycerides, monoglycerides, fatty acids, glycerol, they are combined and triglycerides are resynthesized in the endoplasmic reticulum. Then these triglycerides, they are covered by, these triglycerides are now covered by, they are covered by a coat of beta lipoprotein, beta lipoprotein in the endoplasmic reticulum. And when they are covered by beta lipoprotein in the endoplasmic reticulum, now this is the beta lipoprotein, this is the triglyceride, this is the triglyceride. Now this triglyceride fat, which is covered by beta lipoprotein, it is now known as a chylomicron. It is now known as a chylomicron. And these chylomicrons are released by intestinal epithelial cells from their basal margin into the lamina propria of the villi. I will show it here. I will show it diagram. Suppose this is the villus. This is the villus. This villus is covered by intestinal epithelial cells, brush bordered columnar epithelial cells. It is covered by brush bordered columnar epithelial cells. So these brush bordered columnar epithelial cells, they took micelles in the form of pinosomes. They took micelles in the form of pinosomes. Then these micelles in the endoplasmic reticulum, they generated triglyceride fats. They generated triglyceride fats again. Then these triglyceride fats, they get covered by a coat of beta lipoprotein. So this is the triglyceride fat. It's the triglyceride fat, and it is covered by beta lipoprotein. Beta lipoprotein coat. So it makes this chylomicron. Sorry, it forms a chylomicron. It makes this chylomicron water soluble. Beta lipoprotein coat makes this chylomicron water soluble. Now it is present in the cytoplasm of intestinal epithelial cell. This is the basal margin of intestinal epithelial cell. This surface is the basal margin of intestinal epithelial cell. This is the basal margin. Basal margin of intestinal epithelial cell. So through it is basal margin, these chylomicrons, they are released into the lamina propria. So this intestinal cell, it shows exocytosis, but from it is basal margin. And it releases these chylomicrons in the lamina propria. In the lamina propria, there are small lymph vessels known as lacteals. These chylomicrons can easily pass through this wall of lacteal into it. So chylomicrons, they accumulate in the lymph which is contained in the lacteal. In lacteal, there is the lymph. This is the lacteal. Lacteal is a small lymph vessel. It is a small lymph vessel. And in this lacteal, these chylomicrons, they accumulate. And then they are carried along with the lymph. Into lymphatic circulation. So chylomicrons, they enter into lymphatic circulation. And lymphatic circulation then finally carries them into blood circulation. It transports them into blood circulation. As I have told you earlier, Lymph is formed from blood and it is returned back to blood. So all the lymph vessels of the body, they unite in the trunk region, forming right lymphatic duct and the thoracic duct, which then unite with the superior vena cava and join and pour the lymph into blood, right? So along with lymph, these chylomicrons are carried into blood circulation, and then blood circulation takes them to adipose tissue. Blood circulation takes them to adipose tissues, where they are stored, where this triglyceride fat is stored. So this is how fats are absorbed. So fat absorption is slightly different. It is entirely different from the absorption of amino acids and glucose and other nutrients. So the end product of fat digestion, diglycerides, monoglycerides, and fatty acids, they get surrounded by bile salts and lipoproteins. Sorry, they get surrounded by bile salts and phospholipids. So they form colloidal aggregates known as micelles. Those micelles, they come in contact with the intestinal epithelial cells, right? As they come in contact with the intestinal epithelial cells, they form invaginations, and these chylomicrons, they are deposited in these invaginations. They enter into these invaginations in the form of water-soluble droplets. So these invaginations, they cut off in the form of pinosomes. Invagination is cut off in the form of pinosomes. And then these pinosomes, they join with the endoplasmic reticulum. And then endoplasmic reticulum may transport them to Golgi body, where Golgi body secretes beta lipoprotein covering over these. In endoplasmic reticulum, diglycerides, they combine with fatty acids to form triglycerides. Monoglycerides combine with two fatty acids to form triglycerides. Glycerol combines with three fatty acids to form triglycerides. So in endoplasmic reticulum, triglyceride fats, particularly in SCR, smooth endoplasmic reticulum, triglyceride fats are resynthesized. Then SCR may transport them to Golgi body, where Golgi body secretes a protein covering over these triglycerides. This protein covering is lipoprotein, beta lipoprotein. So when triglycerides get surrounded by this beta lipoprotein, they become water soluble. And this water soluble triglyceride fat, it is known as a chylomicron. Then intestinal epithelial cells, they release these chylomicrons by exocytosis through their basal margin, into lamina propria, into the connective tissue of this villus. The connective tissue of this villus is known as lamina propria. Lamina propria. And from lamina propria, these chylomicrons, they enter into small lymphatic vessels known as lacteal. They enter into lacteal. So they enter into lymphatic circulation. And lymphatic circulation carries them into blood circulation, and blood circulation deposits them into adipose tissues, right? So this is how fats are absorbed. So this is all about the absorption of nutrients from the lumen of small intestine. I think this is sufficient for today. Thank you.